All right, hello BIO178. Welcome to our week 4 lab. And in this video, I'm going to give a little bit of background about the particular data set that we are working with for this lab. So first of all, I have to say thank you to Dr. Ballerini, who's pictured right here. She's another professor in the biology department, and she was generous enough to give us this data set to work with so you don't have to constantly think about snails that are related to my research. Her research focuses on different types of plants and specifically what type of genes are related to phenotypic differences in these different types of flowering plants. And the data that we're working with today is not published yet, but this is a working draft title for the work. So again, it was very nice she was willing to share this with us. It's not even published yet. And there are a lot of words here that we don't need to worry about, but the gist of it is what they are looking at in this study is looking at highly divergent regions of the genome. So that means regions of the genome, certain markers that are highly genetically different. And specifically, they're comparing two different species of columbine. Columbine is a type of flowering plant. And what's interesting is that these two species are actually adapted to different animal pollinators. So I'll talk a little bit next about how that might be playing a role in how these plants differentiated. Okay, so specifically with this data set, we are looking at two different types of columbine, Aquilegia formosa, which is pictured here on the left, and Aquilegia pubescens, which is pictured here on the right. So if you look at these two flowers, what do you notice that's different about them? Yes, I really want you to think about it and answer, so I'll pause and give you a minute. You can talk to me if you want. It's fine. I'm pretending I'm talking to someone right now too. Okay, so one of the things you might notice is that, first of all, the flower orientation is different. So Formosa is facing down and Pubescens is more facing up. And you also may have noticed that they're different colors. So Formosa here on the left is red, Pubescens on the right is white. And what's interesting is that, like I said, these two different flowers have different pollinators. This one on the right is pollinated by a hawk moth. This one on the left is pollinated by a hummingbird. And just like many other plants that are pollinated by hummingbirds, they have these bright colors to actually attract the hummingbirds. So that's really interesting. We have these different closely related species, but they look very phenotypically different. So for me, what I'm always interested in and what Dr. Ballerini focuses on too are what are the actual molecular genetic differences that could be causing these big visible phenotypic differences between the species. And so in order to get at that question, what Dr. Ballerini did is collect about 400 plants from across the Sierra Nevada and the coastal California. So here, this is an image. Again, we have our two different flowers on the left, and on the right-hand side is a sampling map where Dr. Ballerini got the data that we'll actually be working with today, where she collected those samples from. So there are several sites here in the Sierra Nevada. My head, I think, is cutting off this legend here, but you have this in your written instructions as well. But the red dots are all places where she collected a Formosa, and the blue dots are where, the black dots are where she collected a Pubescens. So overall, across these 9 different sampling sites, here's a zoomed in version here. That's where the data set we're working with came from. So you might see in some of the data files these abbreviations. Those abbreviations correspond to the different sampling sites. And so specifically what Dr. Ballerini did when trying to figure out what are the potential genetic differences in these two different types of flowers is look at one particular genetic region, which is called the MYB12. And that's the data set we'll be working with today. MYB12 is a transcription factor. And so what that means, as I'm showing here in this image, remember transcription factors are things that bind to promoter regions and then affect and regulate expression of a different gene. If that transcription factor is able to bind, that gene will be expressed, mRNA will be produced. There should be an intermediate right here. That gene produces mRNA, and then the mRNA eventually produces a protein product. And in this case, MYB12 is specifically involved in the pathway to make anthocyanins, which are shown here on the bottom of the screen. Anthocyanins are pigments that cause colors like red, purple, blue to be present in flowers. So remember that one species of flower, Aquilegia formosa, has red pigmentation. So what we're really interested in looking at here in this data set is whether there might be DNA sequence differences in this MYB12 transcription factor. If there are DNA sequence differences, it might affect how well that transcription factor is able to bind to the promoter region, for instance. If that transcription factor can't bind, that gene won't be expressed, then we might not actually get the production of that floral pigment. Remember, the apubescence is white, doesn't have any pigmentation at all. So this is a really interesting data set. Again, this is what I love about molecular ecology, is seeing these phenotypic differences and trying to figure out what the molecular causes are. So this is the data set we'll be working with. Again, big thanks to Dr. Ballerini for providing us this data set, and go ahead and continue with the rest of the lab to find out whether or not this MYB12 gene is different between these two species.